Okay, then maybe that's why we
We had
said
some
quiet.
absentees and stuff today, so
No.
they're just quiet.
All right.
Absentees mean they skip because they're that depressed over how that exam went.
They shouldn't be. I haven't posted their scores yet, so.
That's probably why.
Oh, they had an exam. All
They're
right.
living on the
Friday.
edge.
I threw this in at the bottom. It might have been on there previously. On the bottom of that week 3 repro stuff for your viewing pleasure. This is it's a McGraw-Hill text that and they don't have it in the A&P text or the art, just anatomy and physiology text that they touch on this, but here they dedicate a chapter to development and aging. And so it's a useful review for your purposes of stuff. And I'll try to scroll through based on that handout thing that we looked at last time I was talking to you. That is there. So, and some of it's interesting, but embryology. So it looks at development. Then at the end, it looks at senescence, the aging process. That's part of it. And I don't know that that's something that would be a point of emphasis, but it'd be of some interest. I think the knowledge that you get, it's like answering questions. You know, if somebody asks you a question, it gives you a certain amount of background. Like blood vessels get more taut, they become calcified, they're more likely to form aneurysms. Not in young people, unless there's a pre-existing defect that they're born with, but in older individuals. That's why they have certain kinds of problems are predictable in that regard. That are there, and they do all this stuff. So the definitional stuff, okay? So 16 days old when it consists of the 3 primary germ layers. That's technically when it's an embryo. And then again, I underscore that the ectoderm, mesoderm, and that should be endoderm, but that's just a typo. What's in those particular layers leading up to the stage, pre-embryonic stage, first 16 days. So, and again, when the egg has that window, distal third of the uterine tube. And this is very readable. You know, that's why there's such an immense number of the roughly 300 million. You can see one 1,000 of that number effectively can make it to the destination. Okay. And we know how they're motivated and the little things that are there that go on. Cervical mucus strands, that reverse peristalsis, uterine contractions, and chemical attractants. So all of those are part and parcel. So it's a very interesting process in that regard. It's so as you can see, they get within 5 to 10 minutes, they're in the tube, but then they have to undergo, and this is an important nugget, capacitation. Okay. Plasma membrane of the fresh sperm is toughened by cholesterol, so it prevents the release of those enzymes while still in the male, enzymatic damage to spermatic ducts that are within the male, the ductus deferens. In the female, the fluids remove the cholesterol, okay? The inhibitory factors are diluted, becomes more fragile and permeable to calcium, and the sperm, and as the calcium gets in, it really increases the tail motility. So it's a very interesting, to me, it's always the process, and we look at it more closely in when we get to the reproductive part in the physiology lecture itself about all those different events. Viable for longer than you think, okay, 6 days. Conception is optimal within a few days before ovulation to 14 hours after. So that's that window. And again, that I don't know if they still use it. We used to do when my first wife and I, basal body temperatures to determine what the optimum time for ovulation. Those kinds of, you should take your temperature first thing. And now we have digital thermometers. The old days they had a thermometer that had a lot of increments around, so you could look for tiny incremental raises. Didn't work, but what are you gonna do? Okay, so acrosomal reaction. Okay, now it has capacitation. Once it gets there, then you end up with penetration. Okay. So basically, it's a group effort, as we've talked about before, to get through the enzymes. Again, hyaluronidase big player all over the place, even with toxins, hyaluronidase, because it breaks apart connective tissue, hyaluronic acid. Okay. Then entering bonds with, remember, with the egg, the outer area, the zona pellucida. Okay. Digesting a pass through that. The head and midpiece enter the egg. They're destroyed. And so, indeed, again, the idea of the organelles, including the mitochondria, are all maternal. That's that nugget that I'm sure you've gotten genetically between, total DNA and mitochondrial DNA. It's there, blah, 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 and the haploid, polyspermy. Okay. To prevent it, the fast block, sodium channels, depolarizing the membrane, inhibits more attachment. The slow block, just below the membrane, inflow of calcium, cortical reaction, etc. So there's two mechanisms that prevent multiple, and they give you some illustrations that are part of it. Then the completion of meiosis, don't have to go through that. Now it's a zygote. And then what occurs there in the division into trimesters. This is the interesting part. The idea, I think that's a nugget that's worth remembering. More than half of embryos die in the first trimester. Most vulnerable, as we already know, at that point in time. The second trimester effectively complete most of their development, distinctly human, increases as we get nowadays, as we approach like week 24, the much enlarged chance. A lot of that is due to the fact that we can manufacture surfactant. Surfactant, which we get to when we talk about respiratory, is produced by the type 2 respiratory cells. That is the wetting agent that facilitates expansion and limits rather that the less inflated cells are going to adhere to one another. Okay, and then obviously third trimester, rapid growth, cellular differentiation. And that 5.5 pounds is an important weight measure. I don't know if that would be a freestanding question or not, but we were always taught that when you're looking at that anything under 5.5 pounds, whatever the amount of prematurity, it's a big number. And this, we talked about it a little bit, going through the cleavage stages, implantation, and genesis of the embryo. These occur early on, okay, and so they call them blastomeres. And so by the time it gets to the uterus, 72 hours, now you have the Marulia stage. That's how you pronounce it. Solid ball, still roughly the same size as the zygote because it's getting smaller and smaller. We went over this. Uterine cavity 4 to 5 days, continues to divide, finally into the blastocyst as the zona pellucida disintegrates and releases it. And then you get these layers that are there. I don't know that they're going to get into it, but the trophoblast is going to be really the part that forms the placenta. The blastocele is that internal cavity, and the mass, remember how it's sort of like an asymmetric mass that's inside that hollow ball on one side, on the inner portion, that's going to become the embryo. So and this is where we're going with that. So you can go through all of these different stages and look at that. And you get into the twins nugget and whether they're, dizygotic or monozygotic, different things. Twins are interesting. There's a lot of hereditary aspects to twins. So, and that's when that was something we were cautious of with regard to when my wife was undergoing, at the time, they were using clomiphene and Pergonal. And I think they still use the Pergonal today for trying to stimulate egg production. If either partner has a history of multiple births, that's like the Octomom. Somebody who underwent that, who had a history. And then we had my dad's brother and sister are twins, his grandfather, triplets. So we could have been looking at big time. I would have lost my hair sooner. Not that funny. You'd have to sit in the back of the class. 6 days, implantation. Okay, we're good to go there. Then all of this is effectively what happens to the syncytio is the fusion of the cells, and effectively what goes on. This is all part of the implantation process. Okay, takes about a week, completed about the time the next menstrual period we've started, had in the absence of pregnancy, and this is where we accumulate. And they used to. This was one of the things that they used to do earlier than amniocentesis. They used to use intravaginal syncytiotrophoblast sampling to look for genetic anomalies and abnormalities that were there. But I do not know how much that is done today or not. I do not. I don't know if people do amnio and things like that. Because it was a very sad case happened to my it broke up a marriage. They had like a third child, and they found nerve cells in the amniotic fluid, and even though the baby was born, she was born alive, relatively speaking, healthy, but neurologically significant problems that really affected that marriage. Okay, ages ago. Okay, so HCG again, just to reiterate the importance of that, because it circulates in large amounts relatively quickly and effectively plays a role to stimulate the corpus luteum over time to take over the role until the placenta can do so. And obviously that's the agent for early pregnancy detection. Ectopics and tubals, abdominopelvics, again, so you can see a limited amount can be born for that kind of, it's an unusual ectopic pregnancy. And then we begin to see the embryogenesis as we begin to form those layers, okay, that are there, and the different parts. The primitive streak is part of it, etc. And then and again, to me, the take-home point for this is what's coming here. To understand that the mesenchymal cells in the mesoderm, basically connective tissue that's there, and once those layers have formed, roughly at age 16, now we have the embryo. So that's the part. There's the placenta taking over. And we begin to see now, as we get into the embryonic stage, the differentiation process into organs, presence of organs at the end of 8 weeks, roughly. And that's about the time that we get into the fetal stage. We begin to look at that. Not necessarily functional, but the transition at the end of organogenesis. Okay, so effectively into it, it's changing the shape, seeing the C shape, etc., that you're familiar with, and you can buzz through the slides. And I'm trying to do this in the interest of trying to highlight film points. The big number about ectoderm and the association with nerve tissue, I think, is really critical. That's one of those areas. And when you're reading a question, and whether it's a freestanding question or a passage question, and it's talking about birth defects that are neurological and skin, you start to see the relationship that's there. I'm not getting into all that, and yolk sacs and somites, and because we do all this stuff again in advanced phys. So then the endoderm, gut, interestingly, respiratory epithelium, glands, bladder, and urethra. So gut, and we're looking at this area where gonads, mesoderm. So there's a couple of little exceptions, but I think about the oddball things that are here that aren't necessarily skin or nerve, or aren't necessarily you might think of the bladder and urethra as more connective tissue oriented, but that type of stuff. So now you're at the fetus. Bones have started to calcify. Bones all start, with limited exceptions you might remember, from the cartilaginous shell. Ossification are landmarks that we begin to look at, okay, for aging and things like that. It's like seeing the growth plates that are there on an x-ray. So you'll be, that's all informational stuff that might be relevant. Okay. So as you can see, now we're starting to see those types of changes that are there. The predominant size of the head, you get amniotic fluid, etc., yolk sac. Nutrition. So I don't think it's just stuff that would be on it. Okay, that's there. Again, what the progesterone and the estrogen stimulate. The blood flow is interesting. That I'm sure that they're going to get to. This was the chorion. That was the syncytial trophoblast. The chorionic villi is what they would sample from there. So the formation of the placenta from the time of implantation, about 12 weeks, and that's when the corpus luteum stops that are there. So it's the reverse of normal circulation where you have one umbilical vein and two umbilical arteries. So the fetal heart blood flows into the placenta by way of the umbilical arteries that are there. So I mean, it's a little bit different. And so we're exchanging that relationship that's there. So again, you're going to see a bit of a change with regard to these are arteries in the fetal that eventually have the deoxygenated blood, and the vein has the oxygenated blood, that you will see. Returns to fetus by way of the umbilical vein carrying oxygenated blood. Placenta, pictures, shouldn't. So umbilical arteries, low in oxygen, high, again, fetal waste, discharges waste in the placenta, loads oxygen nutrients, returns by way of the umbilical vein. So we went over that. Most of the venous blood bypasses the liver by way of a shunt called a ductus venosus. So we're understand that the lungs are non-functional. The mother's doing all the maintenance work. So you're going to see ductus arteriosus and the ductus venosus are part of it. And it's when these persist at the time of birth that we see alterations. And again, that's another area that you might be looking at. Some of these typical abnormalities, quote unquote, birth defects, whatever the appropriate term is today, that we begin to see. Okay, so this is that. Talked about that in terms of the heart. The foramen ovale, blood going from right to left directly, should close over. If it doesn't, then you have the residual foramen ovale like Chris Letang having that type of a thing. My wife also thinks he's pretty cute. Just saying. I don't know who it's going to be. You're going to have to fight her for Kenny. Just saying. It's a little older with Garoppolo and Brady are still.
Yeah, she can have them.
She's like, keep your hands off picking.
Yeah.
I got you. I'm just going to send you a picture of who he's engaged to. I know. She's not married.
Don't remind me.
What?
Don't remind me.
You're not in touch with your inner cougar yet. Terrible. Okay. Bypasses along the ovale. Here. So, most of the blood directly into the aorta, because we're bypassing the lung. We don't have, so you have what's called the ductus arteriosus. The lungs only a tiny amount of blood for their metabolic needs. Okay? They change dramatically at birth when, as you can see, cut off from the placenta and the lungs expand with air. So you can take a look at here, the ligamentum arteriosum. That's there. You can basically, this gives you an idea of what's going on with regard to the fetal circulation versus the neonatal circulation. So here you can look through this and see this, when they persist, and there's one, I'm sure they have it here. We don't have to go over, called the tetralogy of flow. I think it's capital AFALLOT. That's very famous. It might be worth remembering what that is, the parts that are part of it. All of these things. Things that change at birth, the functionality of a variety of organs, myelination of the nervous system. So, and you commented on that, and we continue to. That's the interesting part. It's like myelination, eventually head control, 3 to 6 months, degree of lower extremity control and the ability to ambulate. average time 12 to 15 months. All those kinds of things. And then, then we go back to the vertebral column, which because of that, we get the original primary, and then the secondary curvature here, okay, in the cervical area, and the secondary, upon ambulation, the development of secondary curvature of the lumbar area. Some of us stay, like me, in that immature state until they're nearly 72. I am. Okay. Yeah. All I can remember, did you ever, Look Who's Talking one that had the late Kirstie Alley and John, did you ever see that movie?
What's it called?
Yeah, it was that
Look
Look Who's Talking. There was a one and two. It was a really funny movie about, and Joan Rivers at the very end, you hear the baby talking, go, "Have I had a day?" She's like, the baby just was born. "Have I had a day?"
What was this about?
It's about a baby.
Long time ago.
Long time ago.
Okay, so wait, like
90s.
90s. Oh, that's not that.
She's gone. I mean, maybe it's
Before
late.
your time.
Not that much.
Look Who's Talking. It's a very funny
I thought we were
movie.
gonna see like 60s or something.
No. It was very, very
90s.
funny.
Okay.
Very. Okay, so I mean, a lot of stuff goes on. Neonatal first 6 weeks, respiratory adaptations. Okay, so I mean, that's the whole idea, that those getting the baby to breathe on its own very quickly. And you'll notice heart rate, respiratory rate, very, very rapid. And over time, it begins to slow down and approach normalcy. The deterioration of these structures that are there. And as you can see, what they do is the deteriorations to some extent become incorporated as the baby develops, like they call it the ligamentum teres, which means round. It's a structure that you see anatomically in the liver. Okay, and another one on the surface, the ductus venosus, the closure of the foramen ovale, etc. So immunologically, the first 6 months are all about the mom. Infants born with near adult levels of IgG, so the mom is covering until we begin to get the early onset. Maybe you guys are taking immunology, covered some of that stuff. So it breaks down rapidly, high enough for 6 months to protect against blah, 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 blah. And really the immunization schedules are based on a lot of that when we're going to start to do things from that standard. Breastfeeding, again, additional protection comes along with it. Thermoregulation, the brown fat plays a role in that. I don't know how that is. Kidneys take a while. We'll get into that when we do the renal, because we look at the developmental, it creates what you're going to see is this gradient called a countercurrent multiplier. It's not there at birth, which is why they just go all the time. Okay. High rate of water loss, more fluid intake, so you can't concentrate their urine. That becomes time. Prematurity, that's the weight. So it's like wait for date, but that 5.5 pound is really significant. And then we look at the failure of the surfactant with respiratory distress syndromes and the propensity, so these are all problems that are attendant to prematurity. Going again. My voice is going.
Okay, so,
I got to stop drinking.
well, you might want to stop talking. We can give them time to read their passages.
Oh, is it time?
Yeah.
Okay. She's looking at me. All right, you're done. Wait a minute. Immature
Okay.
liver. Go ahead. Yeah, okay. I'll come back on Friday and do birth defects. How's that?
Okay, so there's 2 passages. There's like 5 questions on each one.